Okay, so this was the lab that you guys were supposed to do. You were supposed to read this part first so that you could get some information and then answer the question. So bacteria cells are prokaryotic. What does that mean? Prokaryotic. Yeah, they don't have a nucleus. That's right. They don't have DNA in their nucleus. Very good. Okay, so bacteria are not always bad, and it gave you some examples of when it's good, like when. What are bacteria used for? Yeah, they're in medicines and in foods, and there's many other examples, but that was one. Now, it told you to click on the microbiology book and to click one of these to find out what you could about it, and these were the things you were supposed to say. So that was the answer to number 3. All right, yes? Okay, so let's move down a little bit further. Now, what is the independent variable? What is the independent variable in a study? Yeah, that's what you're changing, okay? So what we're doing in this study is we're going to pick one of these bacteria, right? And then we're going to test all of these different antimicrobials or antibacteria, right, on that particular bacteria, right? So what are we changing? Yeah, the type of antibacterial, right? Whatever we're using, okay? So this is the antibacteria agents are going to be your independent variable. And when asked you to list them, it was going to be sterile filter paper, antibacterial soap, household bleach, disinfectant, penicillin, amoxicillin, erythromycin, right? So that was what you were supposed to list for the products. What is the control? Okay, which one is the control? 1 through 7, which one? 1, right? 1 is going to be the control. Very good. What is the dependent variable? Now, the dependent variable is going to be the effect. And remember, the other way to say dependent variable is the measured variable. So what are we going to be measuring? What are we going to be looking at when we do this study? Yeah, we want to know which one of these disinfectants right here causes the germ to grow the least or not, or to be destroyed, right? Whichever one works the best at fighting the bacteria, okay? So that's exactly what we're looking for. All right, so what variables remain constant? So if you look at this situation here, what are we going to keep constant? Well, the one thing we're going to keep constant is this bacteria, right? We're not going to test, penicillin on this one and then amoxicillin on that one. That doesn't work. So you're going to keep the bacteria, whichever one you chose. You're going to keep that constant, okay? You're going to keep the amount of time that you put it in the oven constant. You're going to put it all in the same oven, right? You're going to keep the temperature, right, the same. So these are all the things that you needed to put in there. And I highlighted on several people's paper, and I put plural, meaning it said variables, right? More than one variable to keep constant. All right, hypothesis. I have to say this, y'all. For the love of all that's good in this world, please stop using I think, I believe, it might. Okay? Our hypotheses need to be very, very affirmative. They need to say penicillin will reduce the growth of bacteria the most, or antibacterial soap will reduce the growth of bacteria. However you're going to state it, you need to just state it. Okay? You can say, if I use penicillin, then the bacteria will grow less or will be reduced. You can say that if you want to put it in if then, but don't say, I think, it might, maybe, right? Yes, you have to, it's affirmative. You have to say, this will happen. You don't have to be right. You do not have to be right, but you have to say that's what you think is going to happen without saying, I think. You say, it will, okay? And if you're wrong, just do it again until you're right. All right, what data are you going to collect? All right, so what's going to happen is I put the bacteria, and so today you're going to put the disc in, right? Now, oh, it didn't go. What are you going to be checking once you finish this experiment? Luckily, that won't let you put the same one twice. What are you going to be checking for once you finish? Yes, the size of the bacteria, right? So I want to see what happens. And so what's going to happen is you've got bacteria in this dish, right? And each one of these antibacterials is here. So what you're going to see is an area around it where there's not bacteria, right? It's like the bacteria has moved away or has been killed, right? That's what you're looking for. So the one that has, and it's called a zone of inhibition, right? But what it really just means is the larger the area around that antibacterial where there isn't germ, right, is what you're going to be looking for. So you're going to measure the diameter, right? You're going to measure the diameter of this zone, right, after you finish. Yes? Now, once you do that, what are you going to do? Are you going to add, subtract, multiply, divide, use a formula, or are you going to compare it? You're going to compare it, right? Several people use a formula. There's no math here, right? So there's not really a formula to use. We're just going to measure it and then compare, right? And then we're going to compare. So that's what we needed to do yesterday to get ready. Now today, you're going to do exactly what I just showed you. You're going to put the bacteria in, and you're going to put the put the bacteria, then put the antibacterias. Then you're going to put this one in the oven, right? Turn it on, put it in the oven, and then you're going to go for 24 hours, right? Did I not turn it on? Yeah. Okay. And then I'm going to stop because I don't want you to see what's going to happen. But then you're going to take it out, right? And then you're going to be measuring that zone of inhibition, right? Or that area where you have dropped the antibacterials. And you're going to use this handy dandy ruler, right, to measure that. Yes? You're going to collect your data. So here's the form we're going to use today. You're going to collect your data here, and then you're going to do your analysis. You're going to do a graph. You have two choices. You can do your graph in Google Sheets, right? Or you can do your graph on graph paper. Either way, okay? And just really quickly to kind of go over the graph in Google Sheets, you're going to type your data in to Google Sheets, right? Then you're going to highlight it. You're going to hit Insert Chart, and it's going to come up with a graph, right? And if that's not the graph you want, then you go over here and you change it to whatever graph that you want. And you can also customize it, change your colors or whatnot. Yes? Remember the basics. Your graph have to have title, axis labels, right? Even increments. It's got to have units, all that stuff. Whether you do it by hand or on the computer, it's got to have all of those pieces. Yes? Okay. Do y'all have any questions?